This is the BioCentury show.
The BioCentury show is brought to you by Faron Pharmaceuticals. Faron is developing a novel immunotherapy that could reshape treatment for high-risk myelodysplastic syndromes, especially in patients who have relapsed after standard therapies. Faron's lead candidate, Bexmarilimab, is a first-in-class anti-Clever-1 antibody that reprograms immune-suppressive macrophages and reignites the immune system, making tumors more responsive to treatment. Phase 2 data from the Bexmab study shows that Bexmarilimab can overcome treatment resistance to standard of care aciditine in relapse refractory high-risk myelodysplastic syndromes. This approach could unlock new possibilities for patients and doctors alike.
Welcome to a special sponsored edition of the BioCentury Show. I'm your host, Jeff Cranmer, Executive Editor at BioCentury. And today I'm pleased to be joined by Juho Jalkanen, founder and CEO of Finnish biotech Faron. Faron's lead program is oncology therapy Bexmarilimab, better known as Bex. It's a humanized monoclonal antibody that binds to immune checkpoint target Clever-1. Clever-1 is an immunosuppressive receptor found on macrophages, leading to tumor growth and metastases. Now, Faron has phase 1/2 data at the American Society of Clinical Oncology meeting in Chicago. It's for myelodysplastic syndromes. We'll discuss the readout on today's sponsored edition of the BioCentury Show. Juho, welcome to the program.
Thank you, Jeff. Lovely being here.
Excellent. Well, let's dive right into this. The macrophage checkpoint field has seen its share of setbacks, to say the least. What has gone wrong in the past?
Well, I think macrophages are a very complex cell type to be targeting, but as such an important cell type, they may make up to 50% of a tumor. So you should be targeting macrophages, but they're very, very elusive, and there's different phenotypes to them as well. And perhaps the early on attempts were, I would say, in a way too simplistic, or thought that all macrophages behave the same. But it's really about the macrophage phenotype, the tumor microenvironment it's in. It's a bit more complex than just that. But we've always felt strongly about macrophages and tackling them. We just cannot simply go by and pretend that they're not there, because they are a major problem.
I'm curious how you landed on Clever-1 and why this target more effectively repolarizes macrophages than therapies against other macrophage checkpoints.
So our scientific founders have actually worked 3 decades in cancer biology, leukocyte trafficking, and what we found early on in our academic studies that among the macrophage checkpoints, this is the most important one to tackle. This seems to control basically all the other macrophage checkpoints, while the others cannot control this one. So this seems to be what we call the mothership, the one you really want to target. But it's a tricky target with a lot of binding sites, a lot of natural ligands, so it's not an easy target.
Well, let's go to the data. You're presenting phase 1/2 data from this program, which is, of course, your lead. What does the readout show?
So excellent. We've been conducting now already in the phase 2 part of this trial in higher risk MDS, and we've been treating relapsed refractory MDS in this phase 2. And it's actually one of the worst cancers to have. Prognosis is usually 5 to 6 months. It's basically as deadly as advanced pancreatic cancer. Nothing seems to work. But here we are, we've taken that 5.6 month survival to 13.4 months, so it's completely unheard of. Again, usually you don't get any responses with existing treatments. We have a response rate of 63. It's completely unheard of in such a bad population, and this could be groundbreaking for these patients.
Tell me a little bit more about that. What do you attribute that success to? I mean, that's much higher than the 5 to 6 month prognosis.
Yes, and that's the whole point of this. Our core of existence is overcoming treatment resistance. And we are actually a combination agent. So we reprogram the macrophages, but in MDS, the macrophages have gone wild and they need to be put back in their place or may, behave well. And that's what we do, but we combine with the leading standard of care, which is an HMA, a hypomethylating agent. We make it work again. So we've now proven that these patients that have failed it, now suddenly it works again. So that's kind of the magic trick to it. But in what we do, since we're not a cytotoxic agent, we're very well tolerated.
What do you attribute that being well tolerated to?
Since usually, in cancer, chemo, other stuff, pretty toxic, myelosuppressive, we are only looking for this immune modulation and reprogram of the macrophages, and in MDS, the actual cancer cells. So that's why, because in MDS, you're deeply anemic, neutropenic. We don't make that any worse for the patient. We actually help the bone marrow to recover, so we get the cancer killed without being toxic and allowing the bone marrow to recover even and become more healthier. So this is actually a great impact and improvement for these patients.
Now, you mentioned that the therapy is in combination with standard of care. Do you expect an anti-Clever-1 approach to have monotherapy efficacy, or do you envision this being combined with standard of care in the future, or perhaps with other immunotherapy mechanisms such as PD-1s?
Well, thanks to the great safety profile this drug has, it can be combined basically with anything, which is great for a combination drug. But actually, this drug, to some extent, has monotherapy activity. We've actually treated a number of advanced solid tumors as a single agent. We've shown that this can really modulate the tumor microenvironment and get T cells activated. And when we see that, that leads to a significant survival benefit. So to some extent, this has single agent activity, but to completely get rid of advanced cancer, kill the cancer away, this should be combined with something more cytotoxic and cancer killing.
Excellent. Well, I noticed that you collected biomarker data in the phase 1/2 trial that suggests a correlation with response. What are your precision medicine plans for future trial designs?
Great question. So especially in large heterogenic cancers like be it colorectal, breast cancer, patient selection will be needed because these Clever-1 positive macrophages, they're found in roughly 22% of basically all cancers if you screen biobank material. So in a lot of cancers, this is applicable. But then you will need patient selection. You would, through a tumor biopsy, you would want to see that there are these Clever-1 positive macrophages in the tumor microenvironment. So that's for solid tumors. But here in MDS, where we're, hitting big numbers in response rates, because in MDS, it's a macrophage cancer. The macrophages have gone wild. They're the actual cancer cells. Everybody's basically Clever positive. So here in MDS, we don't need any patient selection.
Excellent. And what's next? What's the next step for the program?
Well, big steps ahead. It's into an end of phase 2 meeting with the FDA, and then starting up a phase 3 confirmatory trial, getting this drug approved and to patients.
Excellent. Juho will be following this one as you make those next moves with the compound, and we appreciate you joining us on this special sponsored edition of the BioCentury Show.
Thank you, Jeff.
BioCentury would like to thank Faron Pharmaceuticals for supporting the BioCentury Show podcast. Faron believes Bexmarilimab's early data suggests that product could offer new hope for high-risk MDS patients with limited treatment options. Learn more at faron.com.